हेलो माय डियर फ्रेंड्स, आई एम काजल, स्टूडेंट ऑफ एमएससी माइक्रोबायोलॉजी। आज मैं आपको अमीनो एसिड्स के बारे में पढ़ाऊँगी। लाइक व्हाट आर द आई विल टेल यू अबाउट द बेसिक concepts, basic things about amino acids. You can tell anything, amino. That amino acids basically contain composed of means containing carbon, hydrogen, oxygen, and nitrogen. So basically amino acids are the compounds which contains carbon, hydrogen, oxygen, and nitrogen. Basically they serve as a building block of proteins and that are composed of amino group. As I told you that the proteins that are composed of amino acids, carboxyl group or hydrogen atom, and another that is a distinctive side chain. What does it mean to say that at one side of it there will be a amino group, other there will be a carboxyl group, other side there will be a hydrogen there, and other fourth will be a distinctive side chain. So basically all the amino acids have same structures, just the differ at their distinctive side chain. Means all these form a structure of amino acids and all they are bonded with carbon atom. So at the center there will be a carbon atom, at one hand will be amino group, other carboxyl group, other hydrogen, and other the fourth that will be the distinctive side chain. Only due the changes will occur at this side chain will examine or will evaluate the structure of different structure of different amino acids. So this is the general structure, like there an amino group, carboxyl, hydrogen, and there is a distinctive side chain. So this distinctive, the changes at this side chain will evaluate or will consider the different form of structure of amino acids, of different amino acids. But there is an exception that always we heard that except proline is the only amino acids which have not the same common structure of amino acids. Rather all amino acids have their basic the upper portion of this amino group, carboxyl and hydrogen have the same, this only the changes at its side chain. Just always remember the upper part of this amino, carboxyl and hydrogen has the same. You will see in all the amino acids upper these things same in all, only the changes will occur that is in side chain. Okay, means the R group or side chain attached to the alpha carbon. Which one will consider as alpha carbon? When all four sides have different side chains, means all four means each side has different each group of side chains have different in each amino acids. So in the simplest case what we say, the side chain is an hydrogen atom, and the hydrogen atom, the side chain, if there will be a hydrogen atom at the side chain, will consider as a glycine. Means the general structure, the three, this one, and all these four side chains will connect with carbon, and that is considered as an alpha carbon. So one more thing, some of the structure we see, some of the structures we see like in this way. So those which attach with the upper one will consider as alpha carbon, this one is beta carbon, and another will attach to, so this center will consider as a gamma carbon. So same we have after gamma, there will be delta, there will be epsilon amino acid. So depending upon the location of the carbon atom at which amino group is attached. So in amino acids, both the amino group and the carboxyl group are attached to the same carbon atom. So however, many naturally occurring amino acids not found in protein but have structures that differ from the alpha amino acids. Okay, so now there you have heard about amino acid, the positive negative charge. Means positive ions and negative charge. What is the some have positive and negative. So before this, I'll tell you about the amphoteric molecule. Amphoteric molecule means those means when an amino acid is dissolved in water, it exists in solution as a dipolar ion or a Zwitter ion. So what is Zwitter ion? So Zwitter ion can act as either an acid, that is considered as a proton donor, or a base, that is proton acceptor. So in this way, an amino acid is an amphoteric molecule. Okay, when it's dissolved in water, it will exist in the solution as dipolar. Okay, and so through which analysis we consider the positive and negative charges. So the Zwitter ion. Now I'll tell you about, means it can act as either acidic or base, through which you can say about the high pH concentration, about the low pH concentration. So rest I'll tell you of through the structure, like how we tell about, okay, there is an isoelectric point which we calculate. So how do we calculate that? So I'll tell you about it. So we have I'll just explain with the structure. Means basically to analyze the isoelectric point, we have one rule that is called Ajubaju rule. Ajubaju rule itself tells, like Ajubaju means my left and right both has the high pH concentrations or a low pH concentration of molecules. At that time, both sides, some other one have high, other one is low, means low. So at that time, the center will consider as a isoelectric point. Means high concentration of hydrogen ion will always show the less pH. A low concentration of hydrogen ion will always tells about the high pH. So whenever we have a high concentration of hydrogen ion, then we'll say, okay, here high concentration of hydrogen ion, that means low pH. That means there will be a charge of positive. And if there will be a high pH, that means low concentration of hydrogen ion, will consider as negative charge. Okay, so I'll tell you about the structure. Now you can see by the structure you can analyze that there is a high concentration of H plus ion. That means there will be a low pH. And at the last you can see there is a low concentration of hydrogen ion. That is, there will be high pH. Ajubaju rule. So see, you can see at the Aju and at the Baju, another one is a low pH, means high concentration of hydrogen ion, another we have a low concentration of hydrogen ion. At the center, there means both will termed as a neutral. So this will consider as a isoelectric point. Means this is the intermediate pH. Means pH is equals to isoelectric point, Pi. So Ajubaju rule, as I told you, the intermediate part will consider as an isoelectric point. So at the higher pH, the negatively charged species will be species predominance. Means the acid-base behavior of an amino acid in the solution. At low pH, the positively charged species predominance. As the pH increase, the electrically neutral Zwitter becomes predominant. And at higher pH, the negatively charged species predominates. So for today, I'm just ending. So thank you so much to every one of you. Have a good day ahead. Thank you. If you like, then kindly share with your friends, or kindly revise all the things, and if you think that I left something, just say it, drop me in a chat box your thoughts. I'll consider all. Thank you.